The title of the practical is Screening of Amylase Producing Microorganisms. Amylase it is produced extracellularly, and this enzyme is very important in relation to industrial applications. We have already learned about the various applications of amylase enzymes, for example, in desizing of fibers. So, wherever the starch is required to be degraded, amylase enzyme finds its application. So, this enzyme it is useful in several industries like detergent industry, batting of hides, that is leather industry, and several other industries like in the making of corn powder, in chocolate, making of syrups for to be used in food additives in baby food. So there are several applications of amylase, and this enzyme is a carbohydrate splitting enzyme, and the carbohydrate that it degrades it is starch. Starch it is a polysaccharide which is made up with glucose units which are linked together, and it forms a long chain of glucose units which is called as amylose. And when such long chains are cross-linked with other chain through uh, 1,6 linkages, then it is called as amylopectin. So starch is composed of amylose and amylopectin, and this amylase enzyme is responsible for the degradation of this starch into glucose units, maltose units, or in the form of dextrins. So dextrins are short chain of glucose. Maltose it is a disaccharide made up with glucose plus glucose. There are several types of enzymes, amylase enzymes, and they are alpha amylases and beta amylases. So alpha amylases are responsible for acting on amylose unit, and it acts randomly breaking down the 1,4 linkages that may lead to the production of glucose, maltose, and dextrine. So it acts randomly. Whereas beta amylases are responsible for acting from the end of amylose units, and it splits maltose units. That means it acts from the end, and it is responsible for the generation of maltose units. So that is the basic difference between alpha amylase and beta amylase. So in general, we'll be looking for the amylase enzyme which is responsible for the degradation of starch and which is being produced by different microorganisms. So there are several bacteria, several yeast and fungi that produce amylase enzyme. So for this practical, the requirement is starch agar plate, sterile starch agar plate. So starch is a substrate for amylase enzyme, and as we are looking for amylase producers which is produced extracellularly, the medium is a substrate containing medium, so it is sterile starch agar plate. Along with starch, it also contains a source of nitrogen and other growth factors required for the growth of microbes. So this starch agar plate, it is spreaded with soil suspension. So say for example, we are interested in isolating amylase producers from a particular soil sample. So that soil sample, it is 1 gram of soil sample, it is dissolved in sterile 10 ml of distilled water. Then the debris material is allowed to settle down, and the suspension or the supernatant, that is zero point 0.1 ml of supernatant, it is spreaded onto this sterile starch agar plate, and then further it is incubated. Now, say for example, you have a culture and you want to determine whether that culture is able to produce amylase enzyme or not. Then, what we can do is that we can streak that culture, bacterial culture, say for example, uh, onto, we can spot inoculate or we can streak it on sterile starch agar plate like a 2 centimeter line. And then further, this plate after a streak, it will be incubated for 24 hours at desirable temperature. And this is the result. That is, you can see that there is a bacterial growth. So these are the two cultures which have been streaked on sterile starch agar plate, and after 24 hours of incubation we can see that there is growth of these bacterial culture. Then this plate is now ready for further detection. So as we know that starch, when it reacts with iodine, it leads to formation of blue color complex. So considering taking the advantage of this reaction, so wherever the starch is present, and if we add iodine, it will react with iodine, starch will react with iodine to form blue color. So presence of blue color indicates presence of starch. Absence of blue color indicates absence of starch. Absence of starch means the there is degradation of starch. Starch is degraded to glucose, dextrins, and maltose. So therefore, there will be no presence of blue color. Starch, if it is present, there will be development of blue color. So the next step is, onto this plate we are going to pour iodine solution. So if when we pour iodine solution, so you can see here, wherever starch is present, it has turned into blue color, and wherever the starch is degraded, there is absence of any blue color. So this is colorless. So in this case, this culture is not producing amylase enzyme, and therefore there is no presence of zone of clearance. Here there is presence of zone of clearance because this organism has produced amylase enzyme that resulted into degradation of starch around the colony, and therefore it resulted into the development of zone of clearance. So this organism is amylase producing bacterial culture. This is how the screening for amylase producing microbes is done. So this is the first part of a big practical that we are going to study: isolation of amylase producing microbes, that is screening of amylase producing microbes. Second, we'll understand how the amylase activity is determined through amylase assay. Later on, we are going to study the various purification methods for protein. Particularly, we'll try to purify this amylase using several purification methods. And at the end, we are going to establish a purification chart for amylase enzyme. So this is the first part. So we have begun with obtaining a culture responsible for the production of amylase enzyme. Then the next part will be production of amylase enzyme by that particular amylase producing bacterial culture. So this is a laboratory scale fermentation. So during this, the requirements are culture that will act as an inoculum and sterile starch broth. So the major constituent of this medium, liquid medium, will be starch, along with the presence of other nitrogen sources and other growth factors. So this culture is inoculated into sterile starch broth containing flask. Say for example, in 250 ml of flask, we have 100 ml of sterile starch broth, to which we have added, say for example, 5 ml of culture or 1 ml of culture containing 10 raised to 6 cells per ml, and then we'll keep this sterile starch broth inoculated with the culture on shaker, or we can incubate it statically at desirable temperature, say for example 30 degrees Celsius for 24 to 48 hours. So after incubation, we'll get the growth of that bacteria in this medium. So as the organism is amylase producing organism, we expect that the organism has produced amylase enzyme extracellularly in the fermentation broth. So now this broth is ready for the harvesting of amylase enzyme. So the first step, as the enzyme is produced extracellularly, the first step is to separate the biomass. So biomass is the insoluble impurity, we can say, which has to be separated from the aqueous solution. So your amylase enzyme is dissolved in aqueous solution. So the simplest method is centrifugation. So here this is centrifuge. So the contents of fermentation broth are taken into the in centrifuge tubes, and then in order to separate cells, the contents are centrifuged at 5000 to 10,000 rpm for about 10 minutes. So as a result of that, the biomass will get settled down, you can see here, and this is the supernatant. So this supernatant, it contains your enzyme, and these are the cells. So this supernatant is then separated from this biomass. So this is how this supernatant, we can call this supernatant as crude extract of amylase. This is the original sample. This is the crude extract of amylase. Now we want to know that what is the concentration of amylase enzyme in this supernatant. So what we'll do, we'll go for measuring the activity of amylase. It is called as assay. Assay is measurement. So we'll call this as amylase assay. So enzyme activity from the supernatant is determined. It is called as amylase assay. It is called as enzyme assay.